Okay, yeah, so we are recording this and we'll post it later for people who have missed it, or also to help us make notes on the feedback that you give. What we want to talk about today is a tissue page. So as you know, in the knowledge portals, we've so far focused on genes, variants, genomic regions, and diseases and traits. Those are the entities that the portals focus on. And we have had tissue specific information in there, but we haven't provided a way for people to really come at the portals with a tissue of interest and find out everything there is to know about it. So that's what we're working towards. And what we want to show you today is an in-progress tissue page. It's extremely in progress. It's not available in the production yet. It's not complete by any means. It's not even completely functional, but it'll just give you an idea of some of the things that we're thinking about. And so Annie will start off showing you part of that page, and then I'll show you another part, and then I'll go on to show you some other types of visualizations that might be useful for thinking about tissue and gene expression in tissues. And we really want to gather your feedback on what types of data do you find useful? How would you like to see them? And so forth. And please jump in at any minute. This is supposed to be a discussion and it's a small group. So you can use the chat, but also just feel free to unmute yourself and say something. That'd be great. So yeah, I think, Annie, let's turn it over to you and you can get started.
Thank you, Maria. Here's the tissue page as it exists right now in development. And I think, Maria, tempered expectations may be a little too much. I think there's a lot already there, that people will enjoy. But we agreed to start with adipose tissue as something we will show you, just because we thought it looked very visually interesting and stimulating. And so we have here the part that I'm going to do to focus on right now is the expression data that we have. So here we have genes sorted by mean transcripts per million in the adipose tissue that's been sampled here. And right away, this is kind of an interesting thing that we thought was cool. Me with a software engineering background, and maybe not the full biological understanding of what underpins this, but there's all these mitochondrial genes. So that stood right out to me. But I think we're going to go one page further to look at some nuclear genes, because I think that's going to yield maybe a fuller picture of what this page is intended to do. But so here we have a plot showing in this violin plot format the distribution of how many transcripts of these genes are found in the tissue type under discussion, which is adipose tissue. So we have some interesting sort of shapes, and this is in logarithmic scale. It kind of gets, it's visually striking, certainly when you put it into a linear format, but I think it for a lot of these, particularly on the very high side, it's more useful to extract sort of visual insights by sorting it into logarithmic scale. But so you can see, here are these distributions. When you mouse over the plot, you will see each individual data point rendered as a circle. And you can mouse over individual circles, although it gets so dense it might be hard to see some of them, but it will highlight siblings within that data set. So this particular data set, identified by its unique code, once you roll over it, you can see, okay, well, are these, sort of less transcribed samples belonging to one data set? And you can see that in some cases they sort of are, or is there kind of a bimodal effect where maybe one data set has all the sort of highly transcribed samples and one data set has the lower ones. But that's the information that's within the plot. You can also filter by collection if you want to only view data sets belonging to certain collections. You can view them all. And you can also impose a minimum sample count threshold. And that can, a lot of these have hundreds of samples in them. So you can see it slightly changes the shape of the distribution. And this is at the data set level. It imposes a minimum sample count on the data set level. We roll
Hey,
down
Annie.
here. Yep.
Is there an option to view like control versus disease, or just filter out one or the other, or have them side by side?
We don't have a control versus disease. That because that would have to be, I think, annotated on the data set level. But that's an interesting thought. I don't think we currently put it this way, on the API that this data is getting pulled directly from, that data isn't present. If it's out there and if it can be aggregated all together on the level of the data sets that we already possess on that API, that would be great to do. And I think that's a question for some of the other developers on our team who handle that, who do more of the data processing. But no, right now I don't think we have like ready to go data for control versus disease.
But we will. I just wanted to jump in and say we were
Oh,
definitely
okay,
planning
that's
to
good.
add those data.
That's good. We got a
Oh,
question in
great.
the chat. I'm going to take a look at that. Similar, yeah, stratify sex, age, BMI, or variable of interest. That's starting to be, I think, yeah, that's interesting. And so that would sort of be, again, we get those demographic data on the data set level, and then we apply that to the whole thing. But so what we do have, and I'll show you kind of what we do have here is you can break this down and get the full picture of all of the data sets and all the biosamples that we are looking at for this particular gene. Here's one of the mitochondrial ones. It's broken down by biosample, which collections this appears in. You can view the data set on the CMDGA site if you want to see that. And it breaks down some statistical measures of central tendency about those data.
When you get a file,
Yep.
like I received the histology file from the vet, but the one of the tabs, the spacing was really like weird. Like each of 52 lines was like almost a full page, and then some were small and some were big. It was like, but there's like very little. If I adjust the spacing to they're all the same, the lines is. Do I have to say that I modified the file? Like, you know what I mean? Like
To who?
when I send it to CSL.
Maria, okay. Yeah, I was just gonna say, I think that's a question for somebody who's not me, which is fine. We all have technical questions. Yes, genetic data coupled to the expression data. We're gonna talk. Yeah. Thank you please keep the questions coming. I'm a developer. I'm obviously more capable to speak more to the development side of things, or in the terms of quality of life improvements to things that you want to see. But obviously on the logistical side with the data itself, I think those are maybe Maria can step in. And we also have gene links. I don't think that exists for the mitochondrial genes, but we have these gene links, which you can go down as far as you want within these these aggregated sets of information for each gene per each tissue type that we're looking at here.
Could you subset by like subcutaneous or visceral, like, or the depots or anything like that?
On this top level, we haven't added that yet. I guess we could. Like if you wanted to filter, only view these genes in subcutaneous or something like that. Like, is that
Yeah.
on a top level? Yeah. We could get all of the biosamples per tissue type, and yeah, Maria, let's talk about that. I think that might be a fun if that's what people want to see.
Great, thank you.
Introduce that. Yep. Maria, is there anything else? Is this what people want to know? Is there more that you think I should address, or is that pretty much? Because it's quite basic. There's not an enormous amount of bells and whistles on it yet, but that's why we're here, was to sort of solicit, I think, filtering by biosample at the top level to see all of the genes filtered to the same biosample might be that might be good. I think that would be certainly very useful.
Yeah. Yeah, I would just mention that we were planning to add lots more data to this, and also, for example, the T-stat, we haven't calculated that yet. That's a measure of tissue specificity, but that will be there too. So Yeah, no, that was great, Annie. Thank you. Any more questions about this particular part of it? Or comments?
I guess it might also be interesting to filter by technology. I think I saw, I mean, just like based on the number of mitochondrial genes, I'm guessing
Mm.
there's a lot of single cell data sets combined with GTEx, which is not single cell. So maybe there's like some implicit bias from certain data sets or technology types.
Yeah, so Josh, I'm sure you know the answer to this. I was surprised to see all these mitochondrial genes so highly expressed. Is that a fact that they're usually just more expressed, or that some tissues have way more mitochondria than others, or?
I think it might be from the technology used for
Okay.
the single cell RNA-seq. Um, I think single cell tends to pick up more mitochondrial genes. Um,
Yeah.
versus like single nucleus or but yeah, it's been a while since I've looked at that.
Sure. Well, if we, I think if there's even a consideration that that might be the case, you know, it certainly sounds like sorting by technology would be, um, you know, would help contextualize this in a meaningful way, right? We're not asking for, you know, every possible like full control panel where it's going to be overwhelming. But if there's something where people go, oh, I want to see that, or I'm curious about that, and it's reasonably within our power to do so, yeah, I think anything that's going to help people contextualize what they are looking at or make refinements that helps them look at something that they want to see, then yeah, then we should be thinking about how we can do that.
Mhm.
All right, over to you, Maria.
Okay. All right, let me share my screen. Okay, so I'm going to go down to the next section of the page. There's only 2 sections at this point, and this is global enrichment. And before I start on this, I wanted to just kind of explain in more detail what global enrichment is, because I think it's a really difficult concept. At least I had a lot of trouble getting my head around it. So I just want to make sure we're on the same page with it. So here's just a example, a totally made up example about what global enrichment is. Basically, the reason to do global enrichment, excuse me, is that it can point to the tissues that are most relevant for a disease or a trait. And so it combines 2 types of data, genetic association data and epigenomic annotations. And these are made by looking at epigenomic features of the genome, like histone modifications, and then making predictions about which regions of the genome are regulatory elements like enhancers or promoters and so forth. So when you do global enrichment, and so this is done over the whole genome. I've just put one, like you can think of this as one chromosome, I guess, but it's done globally. So it's just a comparison of distribution of GWAS hits. So say we have, these are T2D associations in the genome. Say we have 500 of them that are genome-wide significant all across the genome. And here we've annotated enhancers in the pancreas, and say that their total length combined is 10% of the genome. And I know, again, that's totally off the top of my head. I have no idea whether it's remotely accurate. But if those enhancers are 10% of the genome and the T2D associations are randomly distributed relative to them, then we should see 50 associations within pancreas enhancers, right? But if we see significantly more than that, then we say T2D associations are enriched within pancreas enhancers. Similarly, if thymus enhancers are 10% of the genome, but we see 50 T2D associations in them, they're not enriched. And so the idea is that when associations are enriched within an annotation, that implies that variation within a regulatory element that's active in this tissue impacts the risk of disease. So that this is pointing us to the idea that gene regulation in the pancreas is important for type 2 diabetes. So that's what global enrichment is. And we do it with LD score regression method. And so in the portals, we already show sort of the converse of this information on the phenotype page. So for a phenotype, we will show in which tissues and which annotations the genetic associations for that phenotype are enriched. but here we're doing the converse. So we have we start with the tissue and say which annotations for which phenotypes are enriched in these elements in this tissue. So that's a pretty simple display. And these enrichments have a p-value and a fold enrichment. So we're going to, usually the expected SNPs and SNPs are not presented. We're going to replace this with fold enrichment. And then we will, we don't yet show, but we will show which biosamples are, in which biosamples we see this. Any thoughts about that? Do you find this useful?
I think it's interesting. I guess, are you going to do something similar for like tissue or cell type specific enhancers, or I guess that's a little harder to do with LD score regression, but do you have any thoughts about like that kind of data, or how that kind of analysis could be done?
I'm not sure I totally understand you. If we had specific biosamples within this, would that address?
Oh, I'm guessing.
Anything?
Oh, so like instead of all enhancers for the pancreas, for example, just the enhancers that are specifically active in pancreas compared to all other tissues.
Ah, okay. That's a great idea. Yeah, I don't think we've considered that yet. We might have, but yeah, that sounds very interesting.
Hey, Josh, can you say that again? I just caught the tail end of that. What were you suggesting?
Oh, basically looking at like tissue specific enhancers and looking at enrichment, like GWAS enrichment within those. It's going to be a smaller subset of enhancers, obviously. So maybe like LD score regression wouldn't be the greatest method, but
Do you have a method that you're aware of to filter out which enhancers are tissue specific that you could recommend?
I do not. I know that there have been like various things like just regression-based approaches done by people using single cell data, but I don't know off the top of my head.
Because one thing we are working on is for adding signals that overlap enhancers as opposed to tissue specific enhancers. So, but not like at the genome-wide enrichment level. I guess you could do that. I guess you could apply a method to convert each annotation to, yeah, I don't know, we have to think about that. But your question would be to like, do you want to see the global enrichments for those, or do you want to use those more to prioritize signals?
Yeah, more to prioritize signals, I guess.
Okay.
I guess maybe like the tissue specific enhancer thing might be something that Kyle's like portal or database has done, maybe. I don't know.
Yeah, no, so okay, yeah. So then I can talk a little more about if it's more about prioritizing signals. We actually just talked about, so we have a new method that I do not think is on this page yet. That's going to first, for this tissue, it will find all of the signals across all of the GWAS that overlap it, and that is via a method that Kyle has a draft for. It basically sums the credible set posterior probabilities that overlap with that annotation in this tissue. But then we have an extension that we are, again, there's not our knowledge one that's available, but something that will come up with the degree to which that signal is specific to that tissue as opposed to just overlaps. And both of those we hope will be added to this page either in the next month or by June. So that will be coming. But yeah, that would be a different question than the enrichment, which is more about globally speaking, how much does the trait localize to this?
Okay, cool. Thanks.
And actually, one thing I should say here, even here, when we're running like when we're running these enrichments, we do have the ability to add in covariates for other tissues. And I can't remember what we're doing, but there is a degree of jointly fitting the enrichments across multiple tissues and annotations that does to some extent control for this. Like if you're familiar with LD score regression, the baseline model includes a bunch of annotations that are just, background and it regresses those out. And what we're already doing is when you get the biosample enrichment here, so when you see an enrichment, I don't think we have do we have the biosamples if you expand, I guess not yet.
Not
Yeah, we
yet.
do.
No,
But
it's,
when you get, so when you get the enrichment for islets, that is islet enrichment controlled for all of the other cell types in the pancreas. I do not think right now it's like necessarily controlled for all the other tissues, but there is like a Uber annotation as well. That's like, here's this annotation across all of the tissues. So you do get already some degree of specificity in these enrichments, but empirically, it hasn't turned out to matter as much as you would think. I think it will matter a lot more at the signal level than at the global enrichment level. So that's probably more than you asked for, but we are doing, it's like literally we discussed it this week
Yeah.
about adding this feature.
But that's what the focus group
So-
is about. Cool. I hope that was helpful, Josh.
It was, thank you.
okay, so other than this, I thought I would go through some other ways, some other questions you could ask and ways to visualize expression data. And let's see, I didn't want to show that yet. And just see what your thoughts are about different visualizations and so forth. I'm trying to move this Zoom window. Let's see. So no, it's not what I wanted. Here we go. So here's a couple of slides from that were put together by my colleague Parul, who works with Kyle at CMDGA. And I just brought this up to talk about UMAP plots. So I'm sure you've all seen UMAP plots. They're a way of visualizing lots of different data types, but for gene expression, they're typically used to compare the gene expression patterns in different cell types within a tissue. So they cluster the cells by their gene expression, overall gene expression patterns, and you can see sort of subsets of cells and different, so you can distinguish the cell types in these plots. You can also look at the expression of a gene of interest on these plots and see in which clusters it appears. So that's one type of visualization that we are working on adding, actually. Yeah, I should have mentioned that we're working on a different portal that will be a totally tissue-specific portal for adipose tissue. It will be called the Mammalian Adipose Tissue Knowledge Portal, and we're working on it with Evan Rosen and his colleagues. And it'll just be a center for all things adipose. Yeah, Evan refers to the fatterati as the people who are really interested in adipose tissue. So it'll be useful for the fatterati and also just for researchers who are working on diseases where adipose tissue is important.
What's nice about that is, obviously this is a tissue close to the hearts of common metabolic disease. And so we're hoping that, working with folks in Evan's sphere will also help us build out visualizations and infrastructure that will obviously bring through to the knowledge portal. So this is another added bonus to this, this community that we've collected here. So it's good news.
Yeah. Let's see. This is a visualization that's at CMDGA, the Common Metabolic Diseases Genome Atlas. And they have some really nice visualizations where, for example, this one is single cell RNA-seq profiles in specific tissues. And so you can, in this visualization, you can compare the expression of two genes and see where they are co-expressed or not. And you can look at very specific tissue types and cell types rather. And so, for example, here, it looks to me like these two genes are very highly expressed in beta cells. But so there's a lot of information here to explore. Another type of visualization is differential expression under certain conditions. And so here, Josh, this goes to your question somewhat. So these plots are in the knowledge portals. This is CMDKP, and they're under the screen to gene tab. So we're trying to collect, we don't have very many yet, but we are collecting differential expression data sets and representing them in these volcano plots and tables. So here, this is data from the Kidney Precision Medicine Project, and it compares individuals with or without chronic kidney disease. So yeah, we're hoping to get a lot more of disease-specific differential expression information. So yeah, please stop me if you want to talk about any one of these plots and say that it's more important to you or less important to you. Here's another type of plot that shows tissue specific gene expression. So we're just working on this in a different portal that we're developing. But these are T-stat values, so measures of tissue specific expression. And the higher the T-stat, the more specific the expression is to that tissue. And this is a display for one gene. This is its T-stat values across all these different tissues. So that's another relevant type of data that might be good to show on the tissue page. And let's see. Also, just some general questions. These are things that we think you might want to know about a tissue or cell type, but we also want to hear from you about different things. As we were talking about, you might want to look in a cell type and under a particular condition, which genes show differential expression. You might want to compare expression interactively for 2 or even more genes of interest in a certain tissue. You might want to compare results for one gene or for one cell type across 2 different data sets. You might want to sort of do a meta-analysis. Other things we could imagine would be interesting would be to see if there's altered cell type abundance for a particular cell type in a condition, or does the cell type specific gene expression change, or which genes are specifically expressed in that cell type. That is really all I had to show, but I'm hoping we can have more discussions about what types of visualizations really that you use all the time and find most useful. Anyone want to volunteer with what would be your top visualization on a tissue page?
Yeah, or questions or even just half-baked ideas about things you're interested in regarding the relationship of different cell types or tissues to disease.
I think a lot of those visualizations would be really nice and helpful to have. I also had a questions about perhaps having some type of visualization to compare across different data sets to see whether you're seeing similar differential expression. Like you talked about meta-analysis, which would, like I think that would be great, like being able to look across 2 data sets, but even more, maybe more data sets than that too, and being able to kind of look at the, like maybe in heat map form of like log2 fold changes or something like that, where you can see like how that gene is changing in these different data sets that you might have present, maybe perhaps even in different cell types. I know that might be a difficult thing to put forth and a bit complicated, but that would be helpful to have.
Great, thanks. One question I thought of is, as you know, the portals have entirely human data, but would data from other species be useful to you? I mean, I guess primarily mouse, but anything else? What are thoughts on that? Mouse would be very useful, okay. Got 1 vote for mice.
Yeah, I think it'd be useful, especially if you could select like if there's a way to select too, like organisms.
To see one or the other, yeah, or maybe even compare across,
Or-
but
yeah.
yeah, we're for the adipose tissue portal that I mentioned is going to have mouse data as well as human, and we're just now working out the complexities of the correspondence between mouse and human, because there isn't a 1-to-1 correspondence in orthologs, but yeah, I think a lot of people use my mouse data. Other thoughts?
I guess, would you have different strains of mice?
Ah,
Um,
I suppose
Yes,
so. We
once-
haven't. Go ahead.
Yeah, because like once you start going down like different like model organisms, you have like different strains. Um,
Yeah, that hadn't occurred to me. That's probably something we have to start keeping track of as well. Yep. This is your chance to put in your request for your favorite feature or your favorite
There's-
type of data.
There's only one request. There's a very, very high chance it will be implemented. Okay, well, we can
Oh.
also, oh, go ahead.
Sorry, I don't want to dominate it, but like the dot plot, like with the UMAP also having like some type of dot plot, which is kind of a way of looking at the expression as well. So just having a couple of visual, we use both of those because sometimes things that are clear on a UMAP aren't clear on a dot plot and vice versa. So it gives you a little bit more information.
Yes, thank you. Yeah, we are working on that for the adipose tissue portal, and that would be something that would come over to these portals as well. Yeah.
Okay, thanks.
Okay, well, I guess we can end a little early if there aren't other comments and ideas, but as you know, we're always here and we always want to hear from you. So please just email us, help@kp4cd.org. You know how to find us, I'm sure. So we do really want to hear from you. If you have thoughts about the tissue page or about anything else in the portals, please do let us know. Thanks everybody for coming, and we'll see you next time. Bye-bye.
Thanks.
Thank you.